Now I'm very pleased to introduce today's speaker, Dr. Eric Steinmetz. Eric is a principal scientist here at Lucigen, and he has a wealth of experience in cloning and protein expression and has developed many of our innovative products here at Lucigen. And we're very excited to have Eric presenting today's topic, Competent Cells 101, Maximizing Your Success in Cloning, Protein Expression, and Library Generation. Take it away, Eric.
Okay, thank you, Beth, and welcome to the wild and wonderful world of competent cells. Here's a brief overview of our agenda for today. We'll begin by talking a little bit about the two major categories of competent cells, electrocompetent and chemically competent, the workflows that they entail, and when to choose one over the other. We'll discuss transformation efficiency and some of the keys to maximizing that efficiency. We'll talk about different strains of competent cells and some of the genetic properties that make them useful for different applications, such as phage display, cloning of difficult DNA, or protein expression. And finally, we'll discuss some strains that are unique to Lucigen, which allow for production of plasmid DNA or protein without contamination by endotoxin. So there are two major classes of competent cells that differ in how they are prepared and how they are induced up DNA. So-called chemically competent cells are simpler to work with since they don't require any specialized equipment, just ice, a water bath at 42 degrees, and a 37-degree incubator. And they're useful for most basic cloning procedures, but they have a maximal transformation efficiency that is a bit lower than can be achieved with electrocompetent cells, so they're limited in that way. But they're useful for most routine cloning and plasmid propagation. Electrocompetent cells cost slightly more, and they do require an instrument known as an electroporator. The trade-off, though, is that they can achieve very high transformation efficiencies. So these are the cells of choice when large, high-diversity libraries are being made. Electroporation also works best when amounts of DNA you have are very limited or when working with large pieces of DNA. So how does transformation work? Chemically competent cells are made competent to treatment with salts and divalent cations, which shield the negative charges on the cell surface, phospholipids and lipopolysaccharides, and on DNA, allowing then the DNA to bind to the cell surface. Heat shock then enhances uptake of the DNA by the cells. Electrocompetent cells are induced to take up DNA by application of a brief electrical pulse using the electroporator. And the strong electrical field is thought to induce transient formation of pores in the cell membrane through which the DNA can enter. It's very important that the sample and the cells contain minimal salt, which can lead to arcing. And arc is what happens when the charge is dissipated rapidly and somewhat violently without inducing pore formation. Okay, so just comparing the workflow, they're similar, but they differ in some key points for the process of transformation. In both cases, the cells are stored at very low temperatures, -80 degrees, until they are to be used, and then they're thawed on ice. DNA is mixed with the cells, and then in the case of chemically competent cells, the cells and DNA are incubated on ice for 30 minutes to allow DNA to bind to the cells. After that 30 minutes, the cells and DNA are subjected to a heat shock of 45 seconds at 42 degrees, usually using a water bath. And after that heat shock, the cells are reduced to ice for a few minutes. And then recovery medium is added, and the cells are incubated at 37 degrees to allow expression of antibiotic resistance. The workflow for electrocompetent cells is a little different and takes a little bit less time, mainly because there's no half-hour incubation on ice. Once the DNA has been added to the cells, the mixture is transferred to a special electroporation cube, which has metal plates that are spaced a fixed distance apart, usually 1 millimeter. And the sample fits into a slot between those metal plates. And then the cuvette itself fits into an electrode chamber attached to the electroporator. The electrical field is applied, and recovery medium is then added immediately, and the cells are allowed to grow again for an hour to allow expression of antibiotic resistance. So the total time for electrocompetent cells transformation is a little over an hour, and chemically competent takes an additional half hour for that ice incubation. There are a variety of shortcuts that are possible that can reduce these times, but here we're presenting the procedures that are required for maximal transformation efficiency. Okay, so what is transformation efficiency? I've mentioned that highest efficiency is achieved with electrocompetent cells, but what do we mean by transformation efficiency and how is it measured? So very simply, transformation efficiency represents the frequency with which cells take up a plasmid and form colonies containing that plasmid. And by convention, it is reported as colony-forming units, or CFU, per microgram of plasmid DNA. It's measured under standardized conditions using 10 picograms Puc19 plasmid DNA per transformation. And all of our competent cell products carry a transformation efficiency claim, which is determined under these standardized conditions. Now, it's important to understand that the maximal transformation efficiency that is measured with intact plasmid DNA occurs at very low concentrations of DNA, on the order of 10 to 100 picograms per standard reactant. So in this generalized graph of transformation efficiency versus DNA concentration, shown by the blue line, gives you an idea of how increasing the DNA above 10 to 100 picograms actually results in lower efficiency in terms of CFU per microgram. However, for a given transformation reaction, it's possible to produce more total transformants by increasing the amount of DNA up to at least up to a few hundred nanograms. So what does this mean for you? Considering a strain that has, for example, an efficiency of 10 to the 10th CFU per microgram, if you were to transform 10 picograms of Puc19 DNA into that strain, you should get 10 to the 5th colonies. So clearly, if your objective is to produce libraries of very high diversity, say 10 to the 9th, it would not be practical to do thousands of transformations with 10 picogram DNA at maximal efficiency. Instead, what people do who are attempting to build large libraries is they work farther out on the curve, where there's some sacrifice in efficiency that's made, but higher amounts of DNA still drive a higher total number of colonies. And finding the right balance between efficiency and the mass of DNA per transformation that gives you the best yield is something that will vary depending on vector, ligation reactions, and other variables. So a certain amount of empirical testing may be needed. So why is transformation efficiency important? Why do we make a big deal about it? It's one reason is, as mentioned, many customers are using competent cells to produce high diversity libraries, and particularly those using phage display methods. They're interested in generating large, diverse libraries of clones. And so in order to do this, you need the highest possible transformation efficiency, because lower efficiencies are going to require more DNA and more cells to generate libraries of the same diversity. Efficiency can also be important in cases of limiting amount of precious DNA samples or for more difficult or challenging kinds of DNA. So here are some tips for those who are looking to produce high diversity libraries. So efficiency is measured with intact plasmid, but when we're talking about cloning and library construction, this is usually done by ligation of inserts into a vector. So transformation efficiency that are measured with intact plasmid do not translate directly to transformation efficiency that you'll see with a ligation reaction. And since there are many different vectors and each ligation reaction is unique, you may need to do some tests to determine empirically what the sweet spot is. So here are some general suggestions for doing library construction. It's possible to directly electroporate a ligation reaction, but you need to pay attention to recommendations about heat inactivation of the ligase. And this depends on the manufacturer. For some ligase preparations, heat inactivation is recommended, and for others, heat inactivation can actually reduce transformation efficiency. So you need to pay attention to that. Alternatively, and perhaps better, it's to clean up the ligation reaction, to remove ligase and buffer before doing the electroporation. And so this can be done either by using a commercial PCR cleanup kit or doing a phenol extraction and ethanol precipitation. And then recovering the DNA in water at a concentration of 200 nanograms per microliter or higher. So this allows you to add only 1 microliter of DNA per standard 25 microliter electroporation reaction. And it's critical to minimize the volume of DNA in electroporation. So for a standard 25 microliter electroporation reaction, you can use up to 200 or 300 nanograms of ligated DNA and expect to have increasing numbers of colonies up to that level of DNA, but maintain the volume of DNA at 1 microliter. So, and it's also possible to scale up electroporation. When doing that, we recommend that you maintain this ratio of 200 to 300 nanograms of DNA in 1 microliter of water per 25 microliters of cells. And if you're going to do this kind of a scale up, we recommend testing it at small scale before committing all of your ligation product to transformation reactions to see what kind of colony numbers you're going to get. You can do electroporations at volumes of higher than the standard 25 microliters. If you're going to do 50 to 100 microliters electroporation, we recommend using cuvettes that have a little bit larger spacing between the electrodes, 2 millimeters. And this may need some optimization of the electroporation parameters. In particular, you may need higher voltage than the standard recommendations that we provide 1 millimeter cuvettes. One other fact that you may or may not be aware of that's important for transformation is the nature of the medium that you plate the cells on. And it turns out that there are 2 different formulas for the common culture medium known as LB, or Luria broth, 2 common variants that differ in the amount of salt. The first, LB Lennox, has a lower concentration of salt, 5 grams per liter of sodium chloride, and LB Miller has twice as much sodium chloride. And it turns out that this difference in composition can be important for colony growth, and we find that growth is delayed when transformations are plated on the higher salt LB Miller plates, so colonies are smaller for a while. So we recommend LB Lennox for all of our competent cells, with the exception of our ClearColi cells, which I'll be discussing later on. Just a brief word about how we provide competent cells to different packaging formats. Both the chemically competent and electrocompetent cells are available in a variety of formats. Single reaction per tube is a solo, 2 reactions per tube or duos, and 6 reactions tube or 6 packs. And then there are 96 well plates also. It's important to note that we do not recommend thawing and refreezing of cells after you remove an aliquot, say from a 6-pack. That's not the purpose of 6-packs, because thawing and refreezing can actually dramatically reduce the transformation efficiency. We also can provide custom formats on demand to match your particular workflows and liquid handling requirements. Okay, turning to the next topic on our agenda, choice of competent cell strains, we have a poll question. So Beth will take over from here to run that.
All right, so the first of our poll questions today, we would like to know, which competent cell strains do you use in your lab? And you can make more than one choice, so choose Apply. We are looking at general cloning, XL1 blue, JM109 or JM110, DH10B derivatives, DH5 alphas, any phage display work that's going on. We want to hear from phage display people out there if there are any. And then cloning unstable DNA, anybody that's using stable 2s, stable 3s, sure or able strains, or our own Endura strain. Or if you have other strains that you're using, we'd like to hear about. So, it looks like a lot of people using DH5 alphas and DH10B derivatives, and a few using L1 blues as well. A couple of people using other strains, and no stable 2, stable 3 users. Okay, and a person using Endura. That's wonderful. All right. Well, if everybody's had a chance to vote, I think we will call it. Looks like DH5 alphas and XL1 blues might be in the lead. for most pot line in the audience? Okay, thank you everyone. We'll finish there and we'll move on to the next part of Eric's presentation.
Okay, so we divide our different competent cell offerings into 5 categories, reflecting different applications and workflows. General cloning, phage display, difficult or large DNA, protein expression, and unique specialty strains for protein and plasmid production without endotoxin. So we'll touch on each of these in turn, beginning with general cloning and library construction. These are the workhorses for most of the cloning work that we do at Lucigen, eCloni 10G and eCloni 5-alpha. In addition, we have a variant of 10G that's called 10G F'. It's the same strain, carries an F' element, which slightly alters its properties. The F' element allows bacterial conjugation. It enables infection with M13 phage, which some people use to produce single-stranded phagemid DNA. And it also carries an overexpressing allele of the lac repressor, which affects the blue-white screening activity. 10G and 5-alpha strains are similar in their usage. The major difference is that the 10G strain was developed to enable cloning of DNA containing specific methylation patterns that are found in mammalian and some plant cells. So the direct cloning of these DNAs is possible with 10G, but much less efficient in 5-alpha due to specific methylation-dependent restriction systems that are present in this and other common laboratory strains. Apart from that, they have similar capabilities. Both are compatible with blue-white screening for clones that have inserts in vectors. And 10GF', because it expresses a higher level of lac repressor protein, requires IPTG to overcome the repression of beta-galactosidase expression that's used in the blue-white screen. We offer the 10G and 10GF' cells in a wider variety of formats, electrocompetent and chemically competent, whereas 5-alpha we have only available in chemically competent format. And we tend to think of 10G as being every bit as capable as 5-alpha, with the added capability of enabling the cloning of methylated eukaryotic DNA. So it's the strain we usually reach for almost all of our cloning purposes. I want to briefly highlight some of the features and the genotypes of these strains. The genotypes of eCloni 10G and 5-alpha are listed here, and some of the relevant bits of nomenclature are highlighted in red. First, both strains contain 2 mutations that are usually found in strains used for cloning and propagation of plasmids, END-A and REC-A. And the END-A1 mutation disrupts an endonuclease that can contaminate DNA prep and reduce plasmid yield and quality. REC-A1 mutation reduces recombination activity, which allows for stable maintenance of plasmids that have small repeat sequences. FHU and TON-A are different names for the same gene, and it codes a surface receptor that is used as an entry point by the virulent bacteriophage T1. And mutations in this gene confer resistance to T1 infection. So it's really just a protective measure to prevent T1 phage from destroying valuable stocks. The F' element I've already mentioned has several effects on strain properties, enabling bacterial conjugation, infection with M13 phage, and it also carries that overexpressing lac-I allele. This cluster of genes here, MCRA, MCRBC, and MRR, these are the genes responsible for methylation-dependent restriction of foreign DNA. And so the 10G strain has mutations in these alleles, enabling the cloning of methylated DNA. That's the difference between 10G and 5-alpha. And the last thing I've indicated is a prophage of phage 80, which carries the omega fragment of the beta-galactosidase gene. This is what enables blue-white screening in combination with the alpha fragment that may be encoded on a cloning vector. Okay, the next category of strains is phage display strains. So in phage display, proteins are fused to a phage coat protein and expressed on the surface of the phage. And sequential rounds of phage infection allow selection for fusion proteins that have desired properties. So because the multiple rounds of phage infection don't require competent cells, phage display is not dependent on competent cells per se, but where phage display is, where competent cells do come into play in phage display, is in the generation of high diversity libraries. So we offer 4 different phage display strains with the highest transformation efficiency available. So here are the characteristics of the 4 phage display strains, and the 2 most important to pay attention to are the presence of the F' element and amber suppression. So F' is required for entry of M13 phage, and so all of these strains but one carry an F' element, enabling phage infection. And this one strain, MC1061 F- is the same as SS320, but without the F' element. So this strain can't be used for phage display, but it can be used for the generation of phage display libraries in the absence of interference by that F' episome. And the other feature to pay attention to, again, is this amber suppression. So as I mentioned, in phage display, the protein of interest is expressed as a fusion to a bacteriophage coat protein. And in some cases, there is an amber suppressor, an amber stop codon introduced between the gene of interest, the protein of interest, and the phage coat protein. And this enables the expression of either the fusion protein in its entirety in an amber suppressor strain, which can read through that stop codon, or in the case of a non-amber suppressor strain, the protein of interest unfused to the phage coat protein can be expressed on its own. So it's important to pay attention to whether or not you're dependent on amber suppression for expression of your full-length fusion protein. Okay, turning to the next topic, cloning of large and difficult DNA. This is a subject that we've given a lot of consideration at Lucigen. Knowing a bit about the DNA that you're trying to clone and what challenges it may pose can be very helpful in solving cloning difficulties. So the types of problematic DNA that can be encountered include DNA that encodes toxic proteins, promoter sequences, AT-rich or GC-rich DNA, large DNA fragments, and sequences that contain a high amount of repetitive elements. And so depending on the nature of difficulties, there may be different kinds of solutions. In some cases, a competent cell strain may be able to solve the problem. So an example of that is Endura. In other cases, it's really going to require a combination of a vector, specialized vector, and vector host systems. And we have a number of examples of these that at Lucigen that enable the cloning of otherwise unclonable DNA. So Endura is an example of a strain that can be helpful for enabling the cloning of highly repetitive sequences. Even though the E-clone I10G and 5-alpha strains carry the REC-A mutation, there are some repetitive sequences that cannot be cloned in those strains. And in these cases, the Endura strain might be an answer. There is no specific genotype that you can point to that accounts for the ability of Endura to maintain repetitive sequences, but the strain has been found to be particularly useful for maintaining repeat sequences, and especially those that are found in retroviral vectors. For example, Endura has proven very useful in the creation and propagation of lentiviral genome-scale CRISPR knockout libraries, or co-libraries, that were created at the Broad Institute, and you can follow this link to learn more about that. As I mentioned, in other cases, a host strain alone may not be sufficient to enable cloning and stable maintenance. We've developed a number of different cloning vectors and vector host systems that enable the stable cloning of difficult DNA sequences that are otherwise unclonable. And I don't have time today to discuss these systems in detail, but I want to just refer you to a recent webinar presentation on this topic, which you can access at our website. The next topic, the next category of competent cells is competent cells for protein expression. So we can divide these into two categories: hosts that rely on the bacterial phage T7 RNA polymerase for high-level inducible expression, and strains that are useful for inducible or constitutive expression by native bacterial RNA polymerase and bacterial promoters. I'd like to focus today on our T7 host strains. The T7 system is broadly used for high-level expression of dominant proteins in E. coli, and the strain most often used for T7 expression is BL21DE3. The strain has some limitations, especially regarding the expression of difficult toxic gene products, and so we have a couple of different kinds of solutions for those. In BL21, the expression of T7 RNA polymerase is placed under the control of a lactose-inducible promoter with binding sites for the LAC repressor, which keeps the expression of T7 RNA polymerase shut off. And in addition, plasmids for T7-driven expression of recombinant protein have a T7-specific promoter recognized by the T7 RNA polymerase, but also have LAC operator sequences to help keep the target gene from being shut off. So LAC repressor protein is serving double duty here. It's needed both for the control of T7 RNA polymerase itself, as well as for maintaining the target gene in the inactive state. And the host strain by itself produces insufficient repressor protein to maintain this tight control over both of these promoters, which means there's often leaky expression of T7 polymerase and then of the target gene. T7 expression plasmids often carry a copy of the LAC repressor to help keep this under control. However, the delay required for the expression of that plasmid-encoded LAC repressor means that leaky expression of the gene of interest can occur before the cells have even had a chance to establish control and establish the plasmid. This can be a significant problem for genes that are toxic to cells when they're overexpressed, such as membrane proteins, which can overload the cellular transport machinery. So we have two different kinds of solutions. One is a high control BL21DE3 cells that carry a gene that overexpresses LAC repressor in the host cells. So there's always a pool of repressor available to maintain control over both the T7 promoter and the T7 RNA polymerase gene. A second kind of solution is represented by the overexpressed C41DE3 and C43DE3 strains. These are strains that were derived from BL21DE3 but have a reduced level of T7 RNA polymerase activity. This allows them to tolerate the introduction of expression vectors that contain toxic genes. In fact, not only are the C41 and C43DE3 strains better able to establish plasmids encoding potentially toxic products, but they're also better able to survive the onset of the expression of toxic products when the expression is used, and in turn that can lead to better yield of the target protein in some cases. Okay, turning to the last category of competent cells that we're going to talk about today, competent cells for low endotoxin, and we have another poll question for Beth to introduce.
Okay, and our second and last poll question for today is going to be related to endotoxin. So we would like to know what your needs are for endotoxin-free preparations of either DNA or protein. So either you don't worry about it at all, so it looks like 3 folks don't worry about it at all. You can choose all that apply, so if you need DNA and protein preps, choose both of those options. Sometimes we need DNA preps that are free of endotoxin. Sometimes we need protein preps that are free of endotoxin. And at the bottom we have always need DNA or protein preps that are free of endotoxin. So it looks like most people are not too concerned about it, but we have a couple people that sometimes or always need various preps that are free of endotoxin. So that's great. If everybody got a chance to vote, I think we'll finish and show you the results here. And we'll move to the last part of Eric's presentation. Go ahead, Eric.
Okay, so what is endotoxin and why is it important? Endotoxin, also known as LPS or lipopolysaccharide, is part of the bacterial outer membrane, and it's present in protein and DNA preparation made from E. coli unless special care is taken to get rid of it. It's important because it elicits an immune response in mammalian cells, which can affect viability and growth of cells. And the presence is important for DNA or proteins that are destined for use in mammalian systems, so it must be reduced to very low levels for any proteins or other materials destined for therapeutic use or for cell-based assays. And the removal of endotoxin can be a costly and time-consuming process. So ClearColi is a new solution to the problem of endotoxin removal by eliminating it at the source. ClearColi strains are strains that have been engineered to produce lipid 4A as the only LPS-related molecule. And this was achieved by deleting a set of 7 genes involved in LPS biosynthesis and a compensatory mutation that is required to enable the viability of these cells. Lipid 4A completely lacks that polysaccharide core of LPS, and in addition, the lipid proportion is reduced from 6 acyl chains to 4. Most importantly, lipid 4A is not an agonist of the human LPS receptor TLR4-MD2, meaning it does not trigger an endotoxic response in human cells. So that's shown here. This is an assay using a cell line called HEC-Blue, which is engineered to produce a blue product when the human TLR4-MD2 receptor is activated by binding to LPS. So you can see these cells respond to standard LPS purified from laboratory E. coli. However, there is a complete lack of response to lipid 4A in these cells. So we offer 2 different ClearColi strains which are intended primarily either for protein expression or plasmid production. For protein expression from T7 promoter plasmids, we offer ClearColi BL21DE3. So shown here is an example of protein expression in ClearColi versus standard BL21DE3. And you can see that upon induction, yields of this protein are similar in the 2 strains. To the right are examples of proteins that were purified using only nickel affinity chromatography from either ClearColi or standard BL21DE3. And you can see that measurements in endotoxin activity show 97 to 99% reduction in the ClearColi samples. Now, it's important to note that the assay used here to detect endotoxin was the LAL, or limulus amoebocyte lysate assay, which is unlike the human TLR4-MD2 receptor in that it does have some reactivity toward lipid 4A. So the residual endotoxin that's detected here reflects that lower specificity of the LAL assay. Our second ClearColi strain is ClearColi K12, which is intended for plasmid production. So it has the NDA and REC-A mutations discussed earlier, commonly found in cloning strains. The strain produces DNA yield that's comparable to E. coli 10G with virtually no endotoxin. Again, the residual endotoxin detected here is due to the non-specificity of the assay that's used. I need to mention a few important details about ClearColi. As I said earlier, ClearColi is the one Lucigen strain for which we recommend plates containing the higher salt formulation of LB Miller. So this is needed for growth of ClearColi because these cells are more osmosensitive due to the changes in their outer membrane. Because of their multiple mutations, these strains do grow more slowly than their wild-type counterparts. So although the yield of protein and DNA can be essentially equivalent to their wild-type counterparts on a per-cell basis, it will take a longer time to get that number of cells. The LAL assay does have some sensitivity to lipid-4A, whereas the human TLR4 and 2 receptor does not. So LAL does not perfectly reflect the endotoxic response in human cells. Other mammalian systems, other than human, do have different sensitivities to lipid-4A. So it's important to know ahead of time if that's a possibility for other mammalian systems. You can find out more information about ClearColi at this link at the bottom of the page. And then just a couple of other brief topics. We do, as I mentioned earlier, we do provide custom competent cells. This can be one of our strains or a customer-supplied strain made chemically or electrocompetent to your specified volumes and formats in tubes or plates that you prefer. And there are a few examples of samples that we provide listed here, and you can find those at this link. Okay, and that's all I have for you today, and I'll turn it over now to Beth again for any questions that you have.
All right, thank you, Eric. Thank you, and to all our participants as well. And now we'd like to take your questions. So audience, if you have questions for Eric, please use your chat window on the left-hand side of your screen. And remember that the button at the bottom of the chat window should be set to all so that we can see your question. And while we're waiting for those questions to start coming in, I should note that if you have the webinar and recording and a copy of the webinar slides will be on our website in a few days, so keep an eye out for that. Looks like we do have a question coming in here, a couple of them. So the webinar will be posted on the same page of our website that the existing webinars that Eric mentioned are posted on. And it looks like, here, let's see if we can, there we go. So first question, how do you make custom competent cells for non-E. coli bacteria? And I can speak a little bit to this. We do have some requirements about the custom competent cells that we make. They must be BSL-1 organisms. We do not handle BSL-2 organisms in our production facility. Other than that, we can handle, we can custom quotes for a lot of different projects. So if you just want to contact us and give us details of your project that you're interested in, we can send you some information. Second question, do you have any data about the growth rate of ClearColi compared to BL21? Eric, do you have any thought on that question?
I think our measurements have shown it to be about 50% as fast as BL21. So it should form colonies on a plate the next day after transformation, and in liquid culture it should grow about half as fast.
So about 50% in liquid culture.
Mm-hmm.
All right, looks like we have another couple of people typing in some questions. Non-E. coli bacterial cells. So about a 1-hour doubling time, is that about accurate for ClearColi, Eric?
I would have to look back at the numbers, but that sounds reasonable.
We also
do have some data. I think there's some data on the website showing growth curves for the ClearColi versus standard BL21.
Yep, that's a great point, Eric. I do have an application note posted on our website about ClearColi media and some media formulations that work very well for ClearColi. So if you would like to email us, we can send you that link. You can email the techsupport@lucigen.com inbox, or you can check our website. It's listed under the Resources tab on the product page for ClearColi. So next question, what is the protein expression level using SS320 compared to BL21 type cells? Eric, do you have any thoughts about the use of SS320 for protein expression?
I don't have any specific experience with that strain. The only difference I'm aware of that might affect protein expression in BL21 type versus SS320 is BL21 as B strains have a defect in a LON protease that can sometimes play a role in degrading recombinant proteins. So it's possible that BL21 for some proteins would produce a better yield than SS320 because of that. But I don't have any specific information about protein expression in SS320.
I think most often people will use BL21 type cells, so we can check if we have any data internally, but I don't believe SS320 is commonly used here at Lucigen for protein expression. So we usually use our BL21 or other strains that are associated with the promoters that we're using. All right, question from Fernando. Once transformation has worked on the antibiotic selection media, sequencing of the plasmid reveals it's correct, did not lead to protein expression. So how to troubleshoot that lack of expression if we are sure that the plasmid sequencing results are correct? Is there another expression strain that we could try? Do you have any thoughts on that, Eric?
Well, it may depend on the source of the recombinant gene that you cloned. If it's from a non-bacterial host, then it's possible that the codons are not optimal for expression in E. coli. So one solution is to have gene codon optimized. There are strains out there that provide rare tRNAs that can be helpful in some cases for non-codon optimized genes. We don't have any of those strains. So lack of expression is a challenge, and if you know that if it's a non-bacterial origin of that gene, then the first thing to look at, I think, is codon usage.
Okay, great. And then our next question is about Lucigen having a specific competent cell type that's best for both cloning and expression. So one-step transformation for cloning and expression. Do you have any recommendations, Eric?
So actually we have a cloning expression system that uses a bacterial promoter and uses the 10G cells as the host. And that's usually a very effective expression system. It uses a rhamnose-inducible promoter system, which is tightly controllable and inducible and can produce very high levels of expression comparable to what T7 expression does. And that uses the 10G cells as cloning host, so you can clone directly into this 10G strain and then pick colonies and induce expression on them the next day. So that's a system that we call the XPSO system, and the version of it that would be easiest to perform cloning and expression in a single host is the Expresso Rhamnose system.
And that's a rhamnose-inducible promoter, right, Eric?
Right.
Okay. And Matthew, if you would like more information on that, just shoot us an email at techsupport@lucigen.com and we can send you some information. Oh, and there it looks like Glass has popped it into the chat window there. So you can follow that link to find more information about the rhamnose protein expression and cloning system. All right. It looks like that's about all the time that we have, and I think we've answered most of our questions here. Great questions, guys. Thank you very much. For future questions, please keep the conversation going. Email us at techsupport@lucigen.com if you'd like more information on these or any other products. We're happy to help you. And thank you so much for tuning in today. Thank you, Eric, for being a great presenter, and thank you to our audience for tuning in. We wish you the best. We'll sign off and close the webinar now.